Hello everyone. As you all know that we are discussing about the life processes and this is the third video for the life processes. We have started the series of lectures for 10th CBSE biology and the first chapter of ours that we are discussing. This is the third video and we are continuing for the autotrophic mode of nutrition. In the previous video, I have told you what the autotrophic nutrition is and the process which is responsible for the formation of food, that is photosynthesis, the raw materials of photosynthesis. So to see that you can have a view for the video, the previous video, and the link I will share in the description of this video. Now let's come to the experiments which prove that light is necessary for the photosynthesis, which prove that chlorophyll is necessary for the photosynthesis, and which prove that CO2 is necessary for the photosynthesis. As you all know that the equation is what for the process, that is 6 CO2 plus 12 H2O, that is C6H12O6 plus 6O2 plus 6H2O. This is the process which is responsible for the evolving of the oxygen in atmosphere which we breathe in for the respiration, and not only we breathe in, all the aerobic organisms. So this is the byproduct of photosynthesis. That is the another story. Now we are concerned with the prerequisites which are responsible, which are needed for the process. And the prerequisite for them is key. Here what is needed? Chlorophyll is needed. Sunlight is needed. Also for the photosynthesis, the raw materials are what? They are the CO2 and water. Just remember this term by heart, that CO2 and water, they are the raw materials, right? Now over the arrow we used to write for the chlorophyll and sunlight. Chlorophyll is present in the chloroplast of the leaves, and sunlight we get from the sun, which is the light energy. Like through the sunlight we get the light energy which is converted into chemical energy through the process of photosynthesis. Now, chlorophyll is the pigment which is responsible for the trapping of the sunlight. Why I am discussing this here? Because we know that chlorophyll and sunlight is necessary. But through the experiment, we will prove that yes, the sunlight is necessary for the photosynthesis. And in another experiment, we will prove chlorophyll is necessary. And the last experiment, it is there for the CO2. Now, just see here, and first we have to consider some terms here. First is the de-starching. What is de-starching? De-starching is the keeping the potted plant into the dark. In the dark, the plant it is kept for some time. That is the de-starching. Actually, in the dark, when the light is not there, the photosynthesis couldn't occur, and the present starch that will consumed off. So that is what the de-starching is, right? Next term is restarching. Restarching is what? The keeping the plant in the light so that the photosynthesis it occurs, and through the process of photosynthesis, what happened? The glucose is formed. And that glucose, it is stored in the form of starch. So actually, the starch, if it is detected in the experiment, it shows that yes, the photosynthesis occurs, right? So that we have to seek the two terms de-starching and restarching. And let us continue our discussion here. And these terms we will use frequently in the other experiments as well. So first we take a plant and de-starch it. De-starch it means it is kept in the dark for 3 to 4 days so that the starch will get consumed off and the new starch formation will not be there because the photosynthesis is not there. Next, we take the plant and here we just select the one leaf and an aluminium foil, it is wrapped around in the center of the leaf in this way. In this way, I have uploaded two diagrams, right, so that you can better understand. And here the aluminium foil, it is wrapped around in the center, and after this measure we have to restarch the plant again by keeping the plant in light for 3 to 4 days, right? So let's come what happened after the restarching. When the plant it gets restarched for 3 to 4 days, then we plug the leaf. We plug this leaf to which we have put the aluminium foil. After that, the aluminium foil is removed. What we have to done after that, that boil the leaf in water. Why? Just to make the cell membranes break so that it will be more permeable to the iodine. So this is a question. This question can come, why after the plucking of the leaf we boil the leaf in water, and for that you have to give the answer that this is there just to break the cell membrane so that it will be permeable to the iodine. The cells will become more permeable for the iodine. After that, we take the leaf out and we give the alcoholic water bath. Here, this is the water. This big beaker, it is having the water, and this beaker, it is over the flame. And here, this beaker, which is inside this, it is having the alcohol. We couldn't place alcohol beaker directly over the flame because it is a substance, it is a liquid which catches fire. So to prevent this, we give alcoholic water bath because we have to boil the leaf in the alcohol, right? So again, a question is ki, first question it emerges from here that why we give the alcoholic water bath, because alcohol if it directly put over the flame it will catch fire. That is why we give the water bath. And second question is ki why we boil the leaf in the alcohol in this way? And the answer is just to remove the chlorophyll. We have to remove the chlorophyll. Like alcoholic boiling, it remove the chlorophyll. So we give the alcoholic boiling here just to remove the chlorophyll, right? This is the question. And alcohol cannot be put over the flame directly because it will catch fire. So this is the another question here. Now after that, after the boiling, we get the leaf and we just wash it with the warm water. The next step is the we get the leaf and washing it with warm water. Why? The reasons are to remove the stuck chlorophyll, and secondly, to make the leaf soft. You can write to soften the leaf. And after this step, let's come to the last step where suppose this is the leaf which we have washed it with the warm water after, after taking it from the alcoholic water bath. So this is the leaf which don't have alcohol now. And here this region will be detected because here the aluminum foil it is wrapped around in the first steps, so some sort of a scar is there, it should be. Now this is the leaf and we have poured iodine solution over it, and after the pouring out of the iodine solution, what will be the result? The region where the aluminum foil it gets wrapped, the region it will not show the blue black color. Let us take some other color. This region will not show blue black color. Why? Because starch, which is the stored food material, it gives blue black color with iodine. And here, this region, as it is wrapped around through the aluminum foil, here the starch presence is not detected because the starch is not formed. And why the starch is not formed? Because the photosynthesis couldn't occur here, as the light cannot penetrate in this region as the aluminum foil it gets wrapped around. So this is the reason this region will not show the blue black color. The other regions in the sense keep this one and this one, it gets the light, it is exposed to the light, and it will show the blue black color after pouring out of the iodine. So this show that ki, yes, sunlight is necessary for the photosynthesis. Let's come to the next experiment. As we have discussed this experiment as the first experiment, this is somewhat lengthy. Our discussion is somewhat lengthy because now as we continue further for the next experiments, some of the steps they are common. So that will not take the time of yours. Again, now we are about to discuss the experiment to show that the chlorophyll is necessary for the photosynthesis. You have to remember one thing, that it is a pigment. It is a magnesium containing pigment. Pigment is used for the chemical which imparts color, and here the pigment is having the function for the trapping of the sunlight. Why I am discussing this point again and again, because this is an important point for the question. Now for this experiment, we take a plant which is having the variegated leaf. Variegated leaf means the patched leaf, who are having the patches of the chlorophyll. Not the full chlorophyll is present in the leaves. So variegated leaves, they are present in the plant like croton. We all have some of the croton species in our garden. So such sort of plant we take for this experiment, and here what the first step is the de-starching. So de-starching we have discussed with, so you know all key what the de-starching is. After that the second step is the restarching. Again you know all the restarching what it is. Next step is pluck the leaf. Then boil it in the water so that the cell membranes break and it will become more permeable to iodine. Next, alcoholic water bath. Same as for the first experiments, and you should, concerned with the questions which I have discussed. Next is wash with the warm water. Wash with warm water to make the leaf soft, and then pour iodine solution. So what will be the result? The patches which are having the chlorophyll, they will show the blue-black color. Other than the patches which don't have the chlorophyll, they will not show because the starch presence couldn't be detected in those patches. No starch because of the absence of chlorophyll. Right? So this shows that chlorophyll is necessary for the photosynthesis. And let's come to the next experiment which is there to show the significance of CO2. Ki, yes, CO2 is necessary for the photosynthesis, and for that we have to make the experimental setup. But first step is the de-starching, as that of the first experiment we have discussed. Second step here, we have to make an experimental setup like this where the leaf it is fitted in a bottle having the KOH solution in the bottom. In this bottle, in the bottom, the KOH solution is there. This KOH solution is having the tendency to absorb the CO2 of this space because KOH it actually absorbs the CO2. So this leaf it is fitted in this, and here this bottle is having the cork lid which is having a slit. Through the slit this leaf it is in this beaker. Now you have to take care of one point, that this part of the leaf which is in the beaker, it should not touch the KOH. It should be in the vicinity of the beaker only, in the space. And this space it don't have the CO2. Why? Because it is being absorbed by the KOH, right? Potassium hydroxide solution. Now, after that, after this experimental setup, we have to take this setup in the light, and by keeping it in the light, we actually restarching the plant. But in this experiment, we have to take up this whole experimental setup to the light for the restarching, and again plug the leaf, boil in water just to make it permeable to iodine. Also the cell membranes they get break, chemical reaction ceased. Important question it is, why we boil it in the water prior to the alcoholic water bath? Just to cease the chemical reactions. Point to be noted here. Next is alcoholic water bath. We have discussed in the previous two experiments. Next is wash it with the warm water. Right? That also we have discussed. And last step is the pour iodine solution. So as we have poured iodine solution, what will be the result? The region which is inside the beaker, as it couldn't get the CO2 while the photosynthesis during the process of restarching, this region which is inside the beaker, it couldn't get the CO2 because CO2 is being absorbed here by the KOH solution. So in the leaf, suppose if this is the leaf and this region it is placed inside the beaker, so this region will not show the blue black color. Which region will show? This region, which is outside the beaker, as it gets the CO2 from the atmosphere through the stomata present on the surface of the leaf. So this shows that yes, CO2 is necessary for the photosynthesis, right? So these are the three experiments which we have to consider in your syllabus, and the one point which I would like to discuss more here, that is how the plant get the nitrogen. And plants get nitrogen from the soil by absorption through roots as amino acid, nitrate ions, nitrite ions, or ammonium ions. So as we all know that nitrogen is present in the atmosphere, and it is converted into the ammonium ions, nitrate and nitrite, which is called as nitrogen fixation, and this is done through the bacteria. So that is the another story that we will discuss in the nitrogen cycle when we will have the chapter of natural resources and all. But here this question, it is in the form key, how the plant get the nitrogen. So that you will have to remember this point, that it gets through the roots in, and it is present in the soil in the form of amino acid, and it is nitrogen, it is present in the elemental form as N2 form in the atmosphere, and it is fixed. Nitrogen fixation takes place, and these fixed products, it is being used in the form of like amino acid, nitrate ions, nitrite ions, and ammonium ions, and this fixation is being done through the bacteria. So I have planned to cover up the experiments for the photosynthesis that we have discussed, along with how the plants get the nitrogen. So this much is for this video. If you like this video, you have to comment for your queries, what you want as your topics, so that we can make more videos for you. And we are doing the series of lectures, so keep watching. You will get the links in the description of this video. I am planning to make the videos for ninth as well, like this manner only. So just keep watching, share, right? And now we will continue our discussion in the next upcoming video. Bye. Have a great day.